Hi, my name is Mohammed Muzamil Khan. I'm a PhD candidate in the bioinformatics program at Boston University. So today I'll be talking about my poster, Characterization and Integration of Transcriptional and Microbial Profiles of Oral Lesions and Cancer. So leukoplakias, which is a group of oral potentially malignant disorders, have a propensity for malignancy. So when leukoplakias does not have histopathological information, they remain uncharacterized. But when they have histopathological information, these can be grouped as premalignant lesions. And these premalignant lesions, as well as oral cancer, goes through a multi-step process and are thought to be progressing through a series of well-defined stages. So the aim of this particular study is to characterize these lesions using the transcriptomic as well as microbial profiles, which could help us identify some early drivers of transformation. And we also hypothesize that there exist some pathways that drive these lesions as well as cancer towards malignancy. In this study, we had 66 patients' biopsy sample being collected, and we performed total transcriptomics to help us get both host as well as microbial profiles and build a crosstalk model that will help us identify the influence of the microbes on the malignant transformation on the host part. So for this, we have 4 groups which has been identified histopathologically, that consists of normal mucosa as well as a hyperplastic stage called hyperkeratosis not reactive, dysplasia, as well as cancer. So using the host profiles, we first applied signatures from similar studies such as OPMD, which is oral potentially malignant disorders, TCGA head and neck cancer data, as well as special profiles called partial EMT of head and neck cancers. The figure 1 shows GSVA enrichment scores of these studies. For the first two, which is OPMD and TCGA head and neck cancer, we had both up and down regulated signatures. So we took the difference of the GSVA scores between these two scenarios, and that is what's been shown in this top and center box plots. And we see a similar pattern of enrichment that goes from control to the cancer group, along with an enrichment to the PML group, showing that our PML group agrees with the already published data sets and has malignant transforming properties. Partial EMT is a profile that is characterized by severeness as well as metastasis stage of head and neck cancers, and we see again a similar pattern of enrichment going from control to the cancer stage. So this helps us identify that the PML as well as cancer group shows transforming activity as well as metastatic profiles of head and neck cancers. As I mentioned that we did total transcriptomics, this gives us microbial abundance, and in figure 2 I'm showing you here the relative abundance of these microbial taxonomies stratified by each of this histopathological group, starting off from the phylum level, going to the genus level as well as the species level. At the phylum level, it looks very similar across groups except for the cancer group, and to identify which of these genera or species that has been relatively abundant in the cancer group, turns out it to be Fusobacterium and Fusobacterium nucleatum, that is shown to be associated with several cancers, including oral cancers. So using the differentially abundant microbes in cancer as well as PML, we can do a differential expression analysis similar to the one we would do for the human gene part. And the microbe that come as differentially abundant can be checked for its enrichment with the activation of some of these genes. So for this we applied microbe set enrichment analysis that checks for the association of these microbes to the already known interactions with the human genes, and we see that some of these microbes such as Fusobacterium, Campylobacterium, and several others have shown to be positively associated with the activation of these genes such as IF6, I16, and so on, and these could be connected to several of the pro-inflammatory pathways that also come up in our host differential expression analysis and pathway analysis. Similarly, this was repeated for the pre-malignant group, and we saw similar patterns. So this helps us build a crosstalk model and identify these microbes that has a positive influence on the activation of these inflammatory pathways. So in conclusion, I would like to say that our analysis showed significant associations in the PML as well as cancer group along with the immune-related pathways, as well as the host microbial relationships, and these findings will help us understand the common biological mechanisms related to PMLs and their progression towards malignancy. Thank you.